Ay, buti na lang. Recording in progress. Kaya basi pangayuan ko ninyo recording, wala ko ika-provide. Labaw na katong wala ka-appeal. Walay connection. So to begin, we have a share screen muna sa ating lesson for today. Stop share muna tayo. Okay, klaro ba? Wala pa na-change ang PowerPoint. Okay, klaro?
Yes, ma'am.
Okay, thank you. So today, still we're on the first unit of our book. Sometimes it was not followed thoroughly, but a part of the topic was being discussed. So kung meron mang na-miss out na topic, what we're going to do is to fill the gap. So it's your part to read further para yung mga details na wala na mention from the book, ma-catch up ninyo. And hindi kayo ma-missing link. Which is also part of your responsibility as a student. And you must have the habit to read also the book. Para in kaso na mga discussion, ay mga mali, you can intercept, ma'am, merong mali, anyan. So that is a good thing because hindi lang kayo accept ng tama or mali, but you interact. Okay, so part din ng teacher, hindi man porke teacher, she is always right. Baka meron siya na-mention na mali, so part of it. As a student, it is also your responsibility also to determine tama ba yun or mali. Yan. So, hindi porke teacher, siya ay tama tanan. So, part din sa teacher to learn also na makorek. Okay, for today, we're going to have an introduction also to microbiology. And part of the asynchronous na to recap on the past lesson. So, past lesson that you should know how to differentiate what is a pathogen and non-pathogen. When we say pathogen, these are microbes that could cause diseases, while the non-pathogen are beneficial. Like for example, di ba nagainom mo og Yakult? So ang Yakult primarily made up of good bacteria such as lactobacillus. So lactobacillus will help your digestion process in your GI tract. So those are example of beneficial microbes. At the same time, yeast. Yeast is also beneficial because it helps our pastry products, fermentation process, like sa mga beer, sa suka, at sa mga wine, mga cheese. Importante ang yeast. But sometimes, we have natural flora na yeast. We have natural flora body in our yeast. If our immune system is going down, mag-flourish ang mga yeast, especially sa mga babae. That could cause also mga infections sa mga important parts of the body. Like sa oral, we have presented there last week yung oral thrush, which is caused by a certain species of yeast. And also itchy private parts, yeast din yan siya, caused by a yeast. So may negative, mayroon ding positive. Differentiate between the acellular microbes, microorganism, and list several example of each. So as you go through your lesson in a synchronous way, you are able to, at least you have identified something, mga microbes based on ano man ang nasa kusina ninyo. And you have already the basic idea. And list several reasons why microorganisms are important. And explain the relationship between microbes and infectious disease, and differentiate between infectious disease and microbial intoxication. So this will be discussed in today's lesson. Now, during the time that we are young, our parents will always scold us na, "Why do not touch that one kasi may kagaw na," or wash your hand. Wash hands first before you hold your food. kasi makakaong-kaog, kaga or germs. And germs may cause you ill, like stomach pain, or you may have fever. So, yun yung mga basic protective move ng ating, especially ating mga nanay or even our yaya. So, these non-living entities are living organisms called microbes. So, that is the layman's term. Kasi anything that cannot be seen that can cause diseases, we call it microbes. So, microbes means very small. Anything so small that must be viewed with a microscope is considered as microbes. And some other call it germs. Diba? Sa advertisement ng Lysol, germs or yung sa brand, Domex. Diba? Therefore, microbiology can be identified as the study of microbes. Not only yung bacteria alone, but yung mga acellular or non-living components. So microbes said to be ubiquitous, meaning they are virtually everywhere. They are present everywhere. Dili kakalikay. Even you talk, even you grasp some air, microbes are already and invaded our body. Pero yung wala malagi ko nagkasakit. Because you have your immune system who will fight back those microbes that enters your body. Naakay mga cellular components that will clean up, that will fight against those foreign microbes that enters the body. So the various categories of microbes includes viruses, bacteria, archaea, protozoa, even fungi and algae groups. But in other references, microbes were categorized into two major components. We have the cellular infectious agents and the cellular microorganisms. Now, underneath the acellular infectious agents, we have the prions and viruses, apilad po itong viroids. And we have also sa cellular microorganisms. Underneath, we have the two domain cell types. We have the prokaryotes and the eukaryotes. Now, under the prokaryote domain, we have the archaea and the bacteria. And we have the other one, the eukaryotes, algae, fungi, and protozoa. Since microbes man, ang ato ang main topic, wala nalabot dire ang animalia and plantae, but they belong to the eukaryotic domain. Dimensionan diri sa slide is merely more on the microscopic organisms. Okay, so disregard muna natin ito. Kasi we have more time. So if you're going to compare infectious disease and microbial intoxication. Now, if we say infectious disease, these are a result or caused by pathogenic bacteria that occupies our system. While microbial intoxication results when a person ingests a toxin or a poisonous substance produced by the microorganism. Now, we have here sa right part ng slide yung comparison ng infectious disease and their action. Now, infectious disease, a pathogen colonizes the person's body. So like TB. Okay, the pathogen causes a disease. So nag-cause siya damage sa respiratory tract ang TB. Now, the type of disease known as the infectious disease, TB, and another example, we have the MRSA infection or the gas gangrene. Now, while the microbial intoxication, a pathogen produces a toxin in vitro. So, like for example, food poisoning. So, a person ingests the toxin and the toxin causes disease. This type of disease is known as microbial intoxication. Example is staphylococcus food poisoning and foodborne botulism or caused by Clostridium botulinum or botulinum. So, meaning sa microbial intoxication, nakasakit ang tao because of the toxin produced by a certain bacteria. Like what we have, the staphylococcal food poisoning. Kasi ito yung mga anaerobic bacteria. Once they are exposed to the aerobic condition. So anaerobic, saan nato makita ang anaerobic? Makikita natin ang mga anaerobes sa mga vacuum spaced or anything that's no air. So more vacuum pack. So nani sa mga canned goods, nani sa mga vacuum sealed packed food, or anything na nakapack, wala man yung air. Now, once these food products ma-opened or ma-exposed sa air, the bacteria that thrive in that can or pack will be exposed to the aerobic condition or naanay presence of oxygen, and they will die out. Mamamatayan sila. And once they die, they produce a toxin. Now, this toxin, if you're going to ingest this toxin, it will cause poisoning or cause toxication to anything na maka-ingest sa ilaha. So, ano yung reaction? Diarrhea, vomiting, and fever. Yun. Yung component na yun ang nakakos ng illness sa atin. Now, in first microorganism on Earth, no, there's no record actually, but later in 20th century, fossils of primitive microbes, as many as 11 different types, found in Asian sandstone formation in Northwest Australia, date back about 3.5 billion years ago. So ito yung evidence nila. At karoon ng mga rock formations. So these rock formations, so they believe that candidates of the first microbes could be on Earth are the archaea and the cyanobacteria. Because ang cyanobacteria have cell walls. Now these cell walls of the cyanobacteria was preserved through time. Now, microbiology is the study of microorganisms. So microbes, which is visible only in a microscope. So last time we have divided through time scale from discovery era, transition era, golden era, and to the modern era based on the development of the field of microbiology. Now, in the discovery era, we have Barro, Columbella, Girolamo, Fracastorius of Verona, von Plensis, and Kirk. So they have their own theories on the microbes. By Barro, you have disease caused by invisible being. Girolamo, we have living germ caused infectious disease. About kay von Plensis, each disease caused by different agent. And Kirk, finding worms on a patient. Oh, as this theory developed, going to the transition era, by basing on the theory of Aristotle on the spontaneous theory, Anton van Leeuwenhoek developed a microscope. So after he developed a microscope, they have already, or some experiments have trying to prove the theory of the other scientists. So they're trying to prove yung spontaneous theory ni Aristotle. So we have Reddy, Needham, Lazarus, Palanci, and even Pasteur. Now, they tried to disprove the theory of Aristotelian spontaneous theory. Now, transition to the golden era, with the discovery of Louis Pasteur on sanitation or yung process of pasteurization, process of sterilization, and also this discovery of some important vaccine on rabies, there are several scientists follow that kind of step with Louis Pasteur. We have John Tyndall, Lord John Lester, and Robert Koch. So these are the people in the discovery era. Mga dagway nila. Okay, we have Aristotle. Aristotle is not only mathematician, not only an astronomer or philosopher, but also a scientist. So overall, multidisciplinary si Aristotle. Kasi noong panahon nila Aristotle, you must know all. Mapa supernatural, paranormal, or even in scientific process. Ganun ang mga scholar noon. Unlike compared ngayon, specialized na ang mga scholars kung sa yung exact field siya mag-focus. So, Anton van Leeuwenhoek developed a simple microscope. So, ito yung na-develop na microscope by Anthony Leeuwenhoek. So, he already identified some animalcules. So, ito yung mga protozoans identified by Leeuwenhoek using his simple microscope. So, Reddy tried to prove the theory of spontaneous generation. So, he said that there are other organisms that really cause the decomposition of a certain organic matter. And so he proved that by using a meat in a jar with 3 setups. We have the open jar. Ang isang jar is covered with cloth and totally sealed jar. So based on the results, dyan sa baba on every jar. And also, John Needham followed the same setup pero mas improvised using a broth or pinakuloang sabaw using a broth. So parang pareho pa rin ang setup ni Reddy, pero sa kanya he uses boiling. So in his boiling method of the broth or the gravy, mutton gravy yan gagamit. Then Lazzaro Spallanzani used the same setup with Needham, but also at the same time, following the setup of Reddy, yung covering the gravy, then sealed the flask or unsealed the flask. So the gravy is boiled and isa flask is open but not boiled. Then the gravy is katong mga gravy na open, nagkaroon ng microorganisms, and isa ka setup is na-boiled ang gravy pero na-sealed siya, then the gravy is free of microorganisms. So this is a combination of setup of Needham and Reddy. Ay, nawala man ang kay Spallanzani. So comparing the Needham's experiment and Spallanzani, Needham attacked already spontaneous generation on the experiment. So mura gi-revise niya ang setup. But in Spallanzani test Needham. So iyang gi-revise ang test ni Needham but with the mixture of setup ni Reddy. Then we have here Louis Pasteur. Louis Pasteur is considered as the father of microbiology because he's constructed as one neck flask, which improvised all setups coming from Reddy to Spallanzani. He proved biogenesis and disapproved on spontaneous generation. And he also developed pasteurization as one way to sterilize the food. So that's why we have longer shelf life ng fresh milk. We have cheese, we have yogurt. So nag-undergo yan siya that kind of process. And he also developed the germ theory of disease and developed also opposed Darwinism. So hindi siya pro-evolution sa theory ni Darwin. So he is against with Darwin. He developed vaccines for rabies, diphtheria, and anthrax, and more. Nakadevelop siya ng vaccine because namatay ang mga anak sa plague. So Louis Pasteur is a father of medical microbiology. So Pasteur in 1897 suggested that mild heating at 62.8 degrees Celsius or 145 Fahrenheit for 30 minutes rather than boiling was enough to destroy undesirable organisms without ruining the taste of the product. So this is the process called pasteurization. And this is also the theory, a concept followed when we go to autoclaving. Although sa autoclaving, it requires 150 pressure exposed in 15 minutes. Tapos nakalimutan ko lang yung time, murag 95 degrees Celsius. So this is the contribution of Pasteur. So he invented the process of pasteurization, the process of fermentation, the development of effective vaccine, especially in rabies and anthrax. Kasi during that time, nagkaroon ng epidemya sa anthrax, and Pasteur demonstrated the disease of silkworm was due to protozoan parasite. And other contribution of Louis Pasteur, he also coined the term microbiology to aerobic and anaerobic organisms. This proved the theory of spontaneous germination or spontaneous generation. He demonstrated that anthrax was caused by a bacteria and also produced the vaccine against anthrax. He developed live attenuated vaccine for the disease. Then we have here John Tyndall. John Tyndall discovered the endospores or most pathogenic bacteria. And he also suggests that prolonged boiling or intermittent heating was necessary to kill these spores. So in short, if you boil the liquid longer, this is the time where the endospores will be destroyed. So this process is known as tyndallization. So para lang ihalang i-extend katong pasteurization process. Longer exposure to boiling. So that is tyndallization. Kasi noon, hindi man nakadiscover si Pasteur sa mga endospores, si Tyndall man ang nakadiscover sa endospores, and he also discovered for how long the endospores will take or to hold to protect the pathogen. So it needs longer period of boiling. That would be enough time for the endospores to be destroyed. Now we have also Joseph Lister, father of antiseptic surgery. So noong unang panahon, wala pang anesthesia. Ibuhos lang ang ethyl alcohol sa sugat, then mura siya living torture ang surgery noon. So Lister concluded that wound infections were due to microorganisms. He also devised a method to destroy microorganisms in the operation theater by spraying fine mist of carbolic acid into the air. So, although noong unang panahon, this carbolic acid is very poisonous sa atin, karon hindi na ganito ang method. There's an improved protocol already. Pero during that time, acceptable siya kaya no other ways pa man ang mga scientists noon. Murag ito yung pinaka-closest way na to prevent the microbes to infect a certain wound during surgery. Kaya through them, nag-improve yung sistema. Robert Koch, he demonstrated the role of bacteria in causing a disease. He perfected the technique of isolation of bacteria in a pure culture. So he used gelatin to prepare solid media, but it was not an ideal because some noon broth ang gamit, hindi pa agar-agar. Since gelatin is a protein, it is digested by many bacteria capable of producing proteolytic exoenzyme, gelatinase, that hydrolyzes the protein to amino acids. Since he wanted to study the bacteria na with their own position, hindi nag-travel, hindi nagagalaw, so, kailangan niyang gumamit ng gelatin. That is the reason yung gelatin nagamit siya. But I think hindi siya nakadiscover sa gelatin. Ang assistant ni Koch, si Van Elschimus Hesse. So, one of the assistant of Koch proposed to use agar in a culture media para pag-observe sa bacteria, dili na mulihok. So agar is better than gelatin because of its higher melting point. But ang gelatin kasi gikan man po na sa agar. Mas dito man po na gikan ang gelatin. Pero mas processed ang gelatin compared sa agar. So agar has a high melting point of 96 degrees Celsius and solidifying to 40 to 45 degrees Celsius point. So mas madali mag-solidify ang agar compared sa gelatin. And we have Richard Petri. He developed yung Petri dish na gamit natin ngayon sa laboratory. So it is a round container with a glass. Ngayon meron ng disposable, meron ng para siyang plastic type. Wala siya glass pero plastic siya. So it's a disposable siya. So it is a container used for solid culture media. So noon, it is made up of glass. It has a cover and a base plate. Now we have Edward Jenner, who used the vaccine for smallpox. So he discovered the technique of vaccination. Na-remember na ako ang isa ka Korean novela na nahuman ako last week is more on valoration, and that was used to prevent smallpox. They used yung pus ng cowpox, then mag-create sila ng wound sa tao. Then they will introduce yung pus ng cowpox sa tao as a vaccine. But I don't know kung siyang method sa smallpox. Okay. Alexander Fleming, he discovered the penicillin from Penicillium notatum. This is a wrong way to write the scientific name. So dapat this one should be written in bigger letter. The rest are small. So Penicillium notatum that destroy several pathogenic bacteria. So during the discovery of Fleming, giyera to. Sumurag nawad-an na sila og alcohol and daghang mga sugatang soldiers. So most of the time soldiers die to infection. Paul Ehrlich, he discovered the treatment of syphilis by using arsenic, and now arsenic is no longer used kasi poisonous din ito. So he studied toxin and antitoxin in quantitative terms and laid foundation on biological standardization. So ngayon, na-improve na yan sila. We don't use anymore the method of these scientists, but through them, we are able to improve the medicines. Like for example, kay Robert Koch, ang anthrax na vaccine caused by Bacillus anthracis. We have many to mention. Hope you find these people in your assignments. So nasulat mo siya sa inyong notes. So daghan kaayong mga scientists. We have also important discoveries of bacteria like leprosy, bacillus. We have Hansen, Neisseria, gonorrhea. Kaya napangalan ang gonorrhea, Neisseria gonorrhoeae kay Neisser, ang genus name ng gonorrhea. Ogston, we have Staphylococcus, Cilioflor, Diphtheria bacillus, Rox and Yersin, Diphtheria toxin. Sa mga viruses naman, discovered during 18th century, we have Bejarik coined the term virus to a filterable infectious agents. We have Pasteur also developed rabies vaccine and also cultivation of virus into chick embryos. Charles Chamberlain, one of Pasteur's associates, constructed a porcelain bacterial filter. We have also Twort and Herel, who discovered the bacteriophage, in which paano ang virus naka-infect sa host cell. So they have designed how the virus, through these bacteriophages, they demonstrated how the virus will infect the host cell. So we have Edward Jenner, vaccination for smallpox. So in the golden age of microbiology, how can we prevent infection and disease? During that time, like these people, we have Semmelweis or hand washing, Lister's antiseptic technique. Sa panahon ni Nightingale, yung proper nursing. Sa kay Snow naman is infection and control and epidemiology. Sa kay Jenner naman, yung contribution during the golden age, the development of vaccine against smallpox. And kay Ehrlich's naman, yung development of magic bullets as a field of chemotherapy. So those are some ways to prevent infection and disease during the golden age. But now, in a modern era, the microbiology gave birth to different disciplines. So from biology, not only animal and botany or zoology and botany, we have also dealt microbiology that focus more on microorganisms. And through underneath some microbiology, we have the microbe-centered disciplines. We have bacteriology, phycology, mycology, protozoology, parasitology, and virology, which focus on specific components. So bacteriology, bacteria and archaea, phycology, algae, mycology, fungi world, protozoology, more on the protozoans, parasitology, parasitic protozoa and parasitic animals. Not only the microscopic ones, but also the macroscopic. Virology focus merely on the viruses. Kaya nga we have right now, scientists have identified yung mga different variants of viruses and also the type of virus. Like we have H1N1, SARS, karon meron ng COVID, different variants ng COVID, Alpha, Beta, Delta, and Omicron. Diba? Through them, they give the identity of the correct virus. Okay, process-centered, we have microbial metabolism, microbial genetics, environmental microbiology. And they have a different field of focus. So that's all for today. And let's proceed on the lecture today. Saan natin check? almost 1 hour ang recap. Now for today, we're going to discuss yung cell structures and the 2 types of cell domains. So all living things, based on the cell theory, all living things are made up of cells. Cells are the smallest working unit of a living things, and all cells come from pre-existing cell through cell division. That is the reason, through this theory, the viruses were not considered as a living organism. Ito yung category. These are the description when to consider that a cell or an organism is a living. Okay, since viruses don't follow this one, the third theory, so di siya galing sa pre-existing cell kasi it is only made up of, it uses a host. So, X siya. Consider na lang siya as a cellular component. So definition of a cell is the smallest unit that is capable of performing life functions. So based sa text, very small po siya. Wala po siya na enlarge. So cell is very, very small. Now examples of cell, sa itong mga cells na nasa ating body, we have the muscle cell, we have the nerve cell, working together with another cell forming into a tissues. But we have also other organisms that live as a one unit or one cell. Like we have here the amoeba, some bacteria, and other protozoans aside from amoeba. So they live as one cell in a very, very large ecosystem. Now we have 2 types of cells. We have this, or we call it the cell domains. These are the prokaryotic and the eukaryotic. Now when we say prokaryotic, before the karyon or before the development of the eukaryotic organism. Now, to describe the prokaryotic, they don't have structures surrounded by, they don't have a nuclear membrane. So the genetic material was scattered within the cytoplasm. No compartmentalization. Walang mga organelles, yung double-bonded organelles. Ang meron lang dyan, you have, in the cytoplasm, you have the DNA, and the ribosomes, and the plasmid. Okay. Ang plasmid kasi nag-vary from species to species. Now, on the cell membrane, they could have either the flagella or the cilia. Okay. Ngayon, sa diagram, pinag-isa siya. Pinag-isa lang siya. Generally, kung cilia. Kung flagella lang. Ngayon, i-combine lang. So, the bacteria could have locomotory organs like the cilia or the flagella. The pilus could be used during asexual reproduction. Kasi ito yung gagamitin to connect with another bacterial cell to exchange the genetic material. Now, in terms of the eukaryotic, mga eukaryotes. Okay. Sa eukaryotic naman, it contains organelles. Naa siya mga different compartments. Muna ilang gitawag na compartmentalization. And mas complex siya compared sa prokaryotes. And at the same time, they're bigger. Since they are complex, bigger din siya compared sa prokaryotes, sa mga bacteria. So makita natin sa previous slides yung sizes, comparing the sizes of all those microbes. Now, we have here 2 types of eukaryotic. Wala lang na-appeal dere ang fungi. But the best known eukaryotic, we have the plant and animal. Now, sa plant, Both of them have cell membrane, but in plants, they have cell wall, but in animals, don't have cell wall. Both of them have mitochondria. They have cytoskeleton. They have the nucleus. They have the endoplasmic reticulum. And also either smooth or rough endoplasmic reticulum. Meron din mga Golgi apparatus. Ang wala lang sa animal, wala lang silang chloroplast. Saan pong chloroplast din eh? Yan, chloroplast. Wala lang ding cell wall ang mga animal cell. Kaya nga ang animal cell, imagine kung natay cell wall, could we run? Could we, can we move our arms or what? Kung natay cell wall, imagine, jointed, muratag naka-shield, muratag, imagine kung may cell wall ta, all of our external features are very stiff, rigid. Kumusayaw ka, mura kag kawayan, tuod na kawayan. Imagine na lang, dili ka kasayaw ng spaghetti pababa or spaghetti patataas like the sex bomb girls if you have cell wall on your cells. So, ang plants, they have a very large vacuole. Although meron man din sa mga animals. But the vacuole seems to decrease because the vacuole serve as the storage. So wala nang ma-store kasi na-use up siya all the time. So usually ang vacuole sa animal cell is not visible. But in protozoans, vacuoles is also very important. Teka, maradili man yata ni mauninan. Okay. So, sa protozoans, vacuoles are also important to maintain cell shape later on. So, this is a typical animal cell. So, careful tayo sa mga position ng mga organelles because organelles are precisely arranged or placed in a certain way. So, dapat si Golgi, doon siya kay endoplasmic reticulum para ang travel ng mga vesicles hindi masyado malayo. So, this is the nucleus. Ito yung mitochondria. Usually, ganito ang pagka-drawing. Ito yung endoplasmic reticulum. There are parts na merong ribosome. So, this is called the rough endoplasmic reticulum. Kung smooth area naman, we have the smooth endoplasmic reticulum. So this is the Golgi apparatus, and the green one is considered as the lysosome. The small one is the ribosome. Lysosome ang green, ribosome. Aside sa lysosome, meron pa yan isa, yung pinakamaliit, ang peroxisome. Hindi yan siya mention sa iba, pero part yan siya sa animal cell, ang peroxisome. Okay? So a typical plant cell, hindi yan mawawala na meron silang cell wall. That is the identifying character structure to differentiate them from the animal cell. At the same time, meron silang chloroplast which cannot be found sa animal. Okay, they have a large vacuole. Mas malaki ang vacuole nila compared sa animal cell. Meron din silang mitochondria, meron din silang nucleus, smooth and rough RER, and also the Golgi apparatus. Wala lang silang peroxisomes, but they have lysosome. Now let's proceed to the function of all cell parts. Okay, first, let's start from the outermost. We have the surrounding cell. Now, all cells are made up of, have a cell membrane. And the cell membrane are made up of phospholipids, double layer phospholipid. So this is the double layer phospholipid. Nag-inatbangay na sila. So the tail part is facing with each other. So the tail part is known to be the hydrophobic tail. And the head part will be considered as the hydrophilic heads. So when we say hydrophilic, non-water loving, so non-soluble to water. Hydrophobic is soluble to water. That is the reason if you're going to take a bath, you will not get dissolved. Kasi naakay hydrophilic and that would repel also water. And also, may naka-embed ng mga cholesterol compounds, mga cholesterol in between the membrane. And also, we have here yung globular part. We have the proteins. We have two kinds of proteins, the integral protein and the peripheral, integral proteins and the peripheral proteins. Asan naman peripheral? Peripheral proteins. Usually, itong mga peripheral proteins, they will not embed totally the bilipid layer. The integral proteins will integrate the double-layered phospholipid. Yan. And also, itong part na to, you have the carbohydrates. Okay. That makes our membrane semi-permeable, which means it doesn't allow immediately the molecules to get in and get out. So, choosy siya. There are certain molecules that are allowed to enter or in and out of the cell. Kasi kung dili siya semi-permeable, dali lang ma-shrink ang cell. Dali lang mawad-an o components ang cell. Dali lang po mag-burst ang cell. That's why dapat semi-permeable siya para one way to protect also the organelles inside. So selective permeable membrane. So the proteins will serve as the receptor site or the channels of other molecules to enter in and out of the cell. In plants, cell wall is not only present in plants, but also in some protozoans and fungi and bacteria. But cell wall on those will differentiate on different components. Mag-matter. Like sa fungi. Fungi cell wall is made up of chitin. While sa plants naman, made up of cellulose, a type of polysaccharide or a type of sugar, carbohydrate, polysaccharide. Then sa sa bacteria, it's made up of peptidoglycan. Now let's proceed sa intracellular part or the inner part of the cell where the organelles are found. When we say organelles, small parts, small organs. Now we have the number one, we have the nucleus. The nucleus is double membrane bounded part. Makita niyo to, double brown patchy siya because the spaces were considered as the pore. The pores will serve as the pathway where the mRNA will go out during protein synthesis. So it directs the cell activities separate from cytoplasm by nuclear membrane. So this is the nuclear membrane in dark blue and contains genetic material. Okay, ito yung chromatin. Ano yung mga giring-giring dire? We have the gene. Then part of the gene, you have the chromosomes. Then part of the chromosomes, you have the DNA. And the inner part, you have the nucleolus, where the protein synthesis happens. So the nuclear membrane part is made up of two membrane, bilayer gahapon. We have the first outer layer. In between them, it is called the perinuclear space. And we have the inner membrane. And in between those membranes, meron siyang mga outlets. We called it the nuclear pore. This will control the exit of mRNA after protein synthesis. Para mabasa ni translation ribosome, rRNA or we call the ribosomal RNA na nasa cytoplasm. So inside the nucleus, we have the chromosomes. The chromosomes, ito usually during before DNA replication or before mitosis or meiosis, hindi ganito ang itsura ng chromosomes. Dili sila in pairs. Usually single yan siya. But during mitosis on the side during mitotic division, they will be held by a centromere, so naging pair na sila. So it is a thread-like structure in nucleus. It carries the genetic information in every section. Yan. So a chromosome, we have the P-arm, the centromere. So this will prepare the chromosome during mitotic division or meiotic division. And the longer part, you have the Q-arm. And the end part consists also the telomeres. Okay. So inside also the nucleus, we have the nucleolus. The nucleolus produces RNA which are used to make all proteins. So dito nangyari yung protein synthesis. And this will separate them from the DNA. So ito yung DNA nasa nucleoplasm. Now let's go out sa nucleus and proceed to the cytoplasm. Cytoplasm is the area where all the organelles are located. It is also the area where the site of initial metabolic processes happens, like glycolysis, the production of ATP. So hindi lang ang production of ATP mangyayari sa mitochondria. It also happens in the cytoplasm. Pero mitochondria produces more ATP compared to the happen in the cytoplasm. So it removes waste, it helps in cell respiration, and converts glucose into energy. So this is the process of glycolysis, yung converts glucose into ATP or energy. And cytoplasm consists also some enzymes that helps the process in the metabolism. Next part. This is very near to the nucleus. So dapat dili ni siya mabulag sa nucleus. So we have the endoplasmic reticulum, which situated near the nucleus. So based on the diagram, we have here the nucleus. Connected to the nucleus, we have the rough endoplasmic reticulum. Then we have the smooth endoplasmic reticulum. The rough endoplasmic reticulum contains ribosomes. May nakadikit na ribosomes. And si smooth, wala. Kaya siya by name, it is called the smooth. Now, rough endoplasmic reticulum synthesizes also proteins after the nucleus have synthesized proteins. So dito niya ipapasa ang katong newly synthesized proteins enclosed in a vesicle. Then si smooth endoplasmic reticulum, primary function niya is more on lipid synthesis. Now to compare further, the RER and SER, so stands for rough endoplasmic reticulum, and SER for smooth endoplasmic reticulum. So RER, synthesized secretory and membrane proteins plus post-translation modification during protein glycosylation. While ang SER is more on lipid biosynthesis and drug detoxification. Other functions like ER provides mechanical support for colloidal cytoplasmic matrix. Kasi yung protein na i-release nila papunta kay Golgi apparatus, ito yung gagamitin for mechanical support. Magiging organelle later on. ER membrane has a permeases and carriers. So meron siyang mga enzymes and some carriers important for the protein transported protein going through the Golgi. It has a major role in intracellular transport of proteins. Secretory proteins passes through RER, SER before it will be received by the Golgi apparatus. SER is more on sterol metabolism, cholesterol synthesis. Yeah. Next organelle, we have the ribosomes. Now, the ribosomes, di ba makikita natin na ay naka-attach sa nucleus, meron din naka-attach sa endoplasmic reticulum, and meron ding free ribosomes found in the cytoplasm. Now, ribosomes consist of two units. We have the large unit and the small unit, or we call the subunits. And ribosomes are made up of RNA. So meron siyang 3 docking stations for tRNA. So tRNA muna yung nag-translate sa mRNA coming from the nucleus. So kani ang mRNA gikan sa nucleus. So when it goes out sa nuclear pore, the ribosome will translate the messenger RNA in order to produce protein. Yan. So this is another way the protein is synthesized. So aside sa protein na nigawas kay Golgi apparatus, meron ding protein coming from directly sa nucleus by the carried by the messenger RNA. So para siyang fragment of RNA. Now, the ribosome that carries the tRNA will translate the mRNA kung saan protein ang maproduce. So, kanya ang protein maproduce after translating the mRNA. So, these are the functions: dock of tRNA to the RNA code, drop off of amino acids, and afterwards generate proteins. Yan. Later on, if we're going to discuss yung aside sa DNA replication, kung ano ba talaga ang central dogma. Yan. Now, if we're going to compare the eukaryotic and the prokaryotic ribosome, kasi present yan siya on all two cell domains. Now, eukaryotic in terms of size, it is larger than the prokaryotic. So, gamay ragid ang ribosome sa mga bacteria. So they contain 70 to 80 types of proteins, while sa prokaryotic, it contains only 53 types. Sa eukaryote, they have 4 types of RNA molecules. So this is how mag-create ng table for comparison. Dapat in each category, may katumbas na description. So, first category, in terms of size. Yan, dapat dito yung size ang first column. So, this is more about the size. Dapat equal siya. Yan. So, this is the one row. This refers to the size. Kasi minsan, if I requested you in your assignment that to make a table of comparison on these components, usually gagawin yung shortcut, one paragraph that deals more on the prokaryote, and another column, another paragraph refers to the eukaryote, without identifying exactly yung category of comparison. Now, if in comparing, see to it, you have the categories. So this is the category of comparison. Yung basis mo sa pag-compare. So first column, you must have the first category, then yung description on each component. Like, in terms of size, first category, so yan ito, eukaryotic is larger than prokaryotic. So, sa prokaryotes naman, they are very small. Okay. Now, types of proteins, number of proteins. And next category. Sige na basta protein. Okay, they contain 80 types of proteins. Ito sa kanya, 53 types of proteins. So still, they belong to one category of description in terms of number of proteins. Ngayon, the fourth one, it refers to the types of RNA molecules. Ano ba ang mga RNA molecules present sa eukaryotes? So they have 4 types of RNA molecules cited as 28 sRNA, 18 sRNA, 5.8 sRNA, and 5 sRNA. Samantala, sa prokaryote, there are only 3 types of RNA molecules such as 23 sRNA, 16 sRNA, and 5 sRNA. Now next, the ratio, protein ratio, the next category. What would be the protein ratio? So, protein ratio sa eukaryote is 1:1, while sa RNA protein ratio is 2:1. Now, where is synthesized? Protein synthesis by this ribosome is inhibited. Mga inhibitors na siya. Inhibited by, or inhibitors, common inhibitors of the ribosome. So, common inhibitors niya is cycloheximide, while sa prokaryote naman is commonly inhibited by chloramfenicol. See? So, ganito dapat ang sa base sa reference na ako nakuha, nawala tong isa ka category. Yeah. So that is how to compare two components. Okay, but kana siya, dili na siya mawala sa other disciplines. Kung maabot na po mo sa human anatomy, ipa-compare na po sa inyo ang either mitosis or meiosis, or pag-abot na po dito sa nursing practices, basig na mo i-compare na two methods. So, to best present in terms of comparison, ganito dapat ang pag-present ninyo in category. Para dali siya ma-recall, at the same time, dali po siya masabtan. O dili siya malimtan. Okay. So I taught you how to compare systematically. Now let's proceed to another organelle. We have the mitochondria. Now, mitochondria is not present sa prokaryotes. Wala sila'y mitochondria. While in eukaryotes, mitochondria is present in all types of eukaryotic cell. Now, mitochondria also carries DNA. Like the nucleus. Ngunit gina-term nila as maternal DNA. So, ito yung ginagamit sa mga maternal DNA na kung dili available ang father, ang nanay ang kani ang gamiton to show relatedness. So, it produces energy. Primarily, the mitochondria, sikat siya pag when you say powerhouse of the cell, mitochondria ang pinpoint. So, dito siya nag-produce ng maraming ATP compared sa cytoplasm. So, pero kailangan man din yung ATP sa cytoplasm para maka-work on properly sa mitochondria. Kasi ang ATP produced in the cytoplasm is very important in this mitochondrial activity like ETC, electron transport chain. So, para makaproduce siya a lot of ATP. At the same time, alam nyo ba na ang mitochondria also carries a component, chemical component, that could trigger cancer. Once the mitochondria contents were leaked out, mag-leak out sa cytoplasm, the cell will not die. Supposed to be our cells will die in the process of apoptosis. Now, once the cell or like component sa mitochondria mag-leak out sa cytoplasm, this will prevent the cell to die out and may cause cancer. So, ito yung isa sa factor na makakause ng cancer. Maski wala kay trace historical lineage on cancer sa imong ancestors, and nagkaroon tayo ng simakulang ng mga development of cancers in our time. Muna yung rason na makakause of cancer. Ang component sa mitochondria makaprevent sa cell to die out. So, katong cell na di mag-die out, mag-continuously multiply siya, and later on maging tumor. Tapos ang tumor pwede siyang malignant or benign. Now, once ang tumor magkaroon ng blood vessels, pwede siya makategorize as cancer cells or cancer na tumor. Cancerous na tumor. So, yun yung development. So, ang cancer, na pwede siya, dili siya by inheritance, pwede siya acquired. So, mura siya switch sa atong balay na switch on, switch off. Once the components inside the mitochondria will leak out sa cytoplasm, that will turn on the cancer growth, cancer cell to develop. So, isa siya sa mga factor. Now, let's proceed naman kay Golgi apparatus. Now, Golgi apparatus or Golgi bodies works cooperatively with endoplasmic reticulum and nucleus because this organelle will receive the incoming vesicular components or yung mga protein enclosed in a vesicle. Now, kaning mga vesicular components coming from endoplasmic reticulum. So inside that, meron mga proteins. And these proteins will be processed first sa cis phase. So dito, i-repack nila tong protein. Then sa trans phase, mag-create na naman, i-enclose naman nila yung repack proteins or they form a secretory vesicle. And these secretory vesicles will turn into another organelle. Like pwede na siya maging mitochondria, pwede siya maging ribosome, or mo-blend in siya sa other organelles. Yan. So, ganyan ang trabaho ni Golgi apparatus. Parang LBC. So, imagine, makareceive siya ng package. Tapos kung ang package na siya yung mga sub-packages, so iyang i-repack, then i-distribute. Yan. So, dapat si Golgi apparatus is not located far away sa nucleus. Dapat located siya near the endoplasmic reticulum para short lang ang travel sa vesicular proteins. Next, we have lysosomes. Now, the lysosomes is primarily perform like an immune system of the cell because they will engulf large particles or foreign particles or mga organelles na nasisira na worn out na organelles. So, this is the digestive plant for proteins, fats, and carbohydrates. It transports undigested material to the cell membrane for removal, and also cell breaks down if lysosome explodes. So this is also the process of cell death or apoptosis. Now, if the lysosome will break out, this will also initiate apoptosis or cell death. Kakainin niya because lysosomes carries enzymes. So this enzyme will eat up other organelles kung ma-break ang lysosome. Or sometimes mangyari yan, natural man din yan. But because para ang cell na mamatay, ma-digest, and ma-replenish para na katong new cell nasi space na an area where to grow. Kasi kung kani siya, old cell will not disintegrate, hindi siya mawala, mura siya og mura siya tumor. Tapos mag-continue pa siya divide. So kung mawala siya with the help of lysosome enzymes, ma-disintegrate siya into minor components or mga protein, carbohydrates na lang siya, and they will be reabsorbed by another cell. Then it also gives a space for the new cell to grow. Yan.
Ma'am?
Yes?
What would cause po, for example, yung lysosome na mag-explode or yung mitochondria kay mag-leak ang mga components niya? What would cause that po? Random lang talaga siya?
Na certain chemical, like mga, katila lang ha, na ay bata na rin nanguhit sa kuha. Okay. What caused yung interesting, no? Pero nana siya sa cell molecular biology. Asa itong topic na ko sa cancer. Okay. Cause siya para mag-leak out ang ang mitochondria kung naay isa ka component na inhibit sa mga chemical factors sa like vices, sigarilyo. Kuan niya tong BCRA, murag dili niya ma-activate ang BCRA para i-clog ang pores ng mitochondria. Once dili maka-close down ang pores ng mitochondria, may tendency katong chemical component in the mitochondria lang dapat mag-leak out, maka-inhibit siya sa apoptosis ng cell. So, every cell may lifespan. Na kung dili siya mag-undergo og apoptotic activity or cell death, tendency mo occupy siya sa space. Then, katong new cell na mag-develop, dili siya ka-occupy dili mag-disintegrate tong old cell, tapos katong new cell, wala siya room where to occupy para magkaroon siya ng cell growth para mo increase iyang volume. So, dili niya ma-perform iyang function. Muna usahay na yung mga tumor, wala siya, isa sa mga development ng tumor. Napay other factor na katong pathway, Merck pathway, taas na ni siya, mura na rin, nana ni siya sa cell molecular biology. Teka ha, ako ipakita ko unsa to ha, nana ni siya sa other PowerPoint. Later on, pahuman na lang ko ani back, lapit naman. Okay lang, Richard?
Okay lang po, na-curious lang.
Okay. Pahuman lang ko ani, later on, balikan na to tong cancer. Okay. So vacuoles are distinct mostly sa protozoans and plants. Now, sa protozoans, their vacuole is more on contractile because this very highly active kasi it will help them maintain its shape because some protozoans don't have cell wall like the amoeba. Once there's an excess water, ang contractile vacuole will expel the excess water para ma-maintain ang shape ng amoeba. So, hindi siya mag-burst. While sa plant, ang vacuole nila para siyang refrigerator, storage compartment. Now, sa animal, aside from storage compartment, performs also to maintain cell shape. Now, sa plants, chloroplast is not present in animals but present siya sa algae group and sa plants. Now, we have different types of chloroplast. Kaya nga, makita nyo sa mga plants, mayroong green, may red, may yellow, but mostly ang pagkaalam nyo, green. So, which is common knowledge to everybody. Pero may kanya-kanyang color, especially sa mga algae group. Na ay golden, na ay red, na ay silver. Yan. Sa algae group na siya. But sa world of plants, we have green as a primary color. primary pigment. Then, meron ding red and yellow. Yan siya. So that's the end of the organelles. And for your asynchronous task, so you will have your individual work in which for the laboratory individual, you have to improvise your own laboratory setup. Now, before we have the actual setup, you have to identify first ano yung mga materials usually used when you have to perform culture media. Ano yung ginagamit sa microlab. One clue, yung si Petri dish. Yan. At saka si microscope. Yan. Ngayon, if you're going to perform in your house, at home, wala naman tayong Petri dish. Ano yung pwede mong i-improvise as a Petri dish? Okay, another clue. Kung nandito ako sa bahay, wala akong Petri dish, ang pwede kong gamitin as Petri dish ay pwede yung lapad na bote or kayo na bahala. Lapad na bote ang sakua. Okay. Ang botelya, mga empty bottles, ang pwede kong gawing Petri dishes. Or kamo na ang susunod. So those are examples sa mag-improvise or innovate. What materials can you serve as the alternative for the actual materials found in the lab? Okay. And for lecture, describe the following microbes. You have the acellular microbes, domain bacteria, and domain archaea. Walang require for submission, but you have to read more about the cellular microbes, the domain bacteria, and the archaea. Now, for the cancer, let's go back to the question ni Richard. So, para mas magalang, mas klaro, na lang ko i-share na PowerPoint. I-open sa na ako para a while. Okay, ka lang ha. You mean, say time check. Please, pabiyak mo. 239. Hintay lang ha. Wait lang.